Hey, good morning, and we're going to get started now. I hope everybody can hear me, and check the volume. Yep, we're good on the volume. So we finished up chapter 24 on metabolism on Monday. We're going to go ahead and move into the bone chapter. New material. New material, a much more interesting topic area here as we start moving into organ systems, bones and muscles. We can start relating to a lot of what we're looking at here in lecture. We're sort of done with the sort of out there, the chemistry and the cellular respiration. We can start seeing things now when we look at bone physiology and muscle physiology and how things work. Plus, we're going to start addressing more and more occurrences of when things go wrong. Because that's what makes healthcare so interesting in all aspects. Is I haven't said it yet, but healthy people are boring, and so not that there's anything wrong. Being healthy is the goal, but as a healthcare provider, there is a certain challenge in trying to help treat somebody and cure them of a condition or a problem. Anyway, let me bring up my PowerPoints. Let's see, and where did they go? Okay. Here we are. Okay, let me bring this up, then I'll go into share screen and we'll, okay. So now let me get back to this, and here we go. Don't mind me talking. Okay, so we're going to start a brand new chapter today. The next unit is going to be bones and muscles. And this and bones, like we looked at it from lab, we're looking at it now from the physiology of how bone tissue works. Why do bones do what they do? So let me start here. Okay. Now the amazing thing about our skeleton is that we have a skeleton in place when we are about an inch long. 6 weeks after conception, we are shaped, like a baby, because we are a baby. An inch after, and we're an inch long. Conception occurred 6 weeks previously. 3 weeks after conception, 2 weeks after we were implanted in the uterus, we have a heart and it's beating. In another 3 weeks, we have a complete skeleton in place made of hyaline cartilage. And that skeleton will gradually ossify, turn to bone, over the next 23 years. Because it takes us about 23 years till our skeleton is fully formed. The cartilage finally disappears somewhere after the age of 20, 21, usually around 23 or 24. I always like to say 23 because it's sort of in the middle there. But we use the hyaline cartilage as the model to build our bone on top of. But we have a fully functioning skeleton in place 6 weeks after conception. We're only an inch long. We're an inch in length, and this is after we've been in the attached in the uterus for 5 weeks. Conception takes place in the fallopian tube, and it takes about a week to get down to the uterus and implant. So we've only been in the uterus for 5 weeks, and yet we have a fully functioning heart and we have a full skeleton in place. And by 23 years of age, we have a completely formed skeleton with no more cartilage, where bone should be. Now, what do our bones do for us? Well, first of all, they support us. They help hold us upright. They allow us to sit and stand. They protect our soft organs. The viscera in the thoracic cavity is protected by the ribs and the spinal cord and the sternum. The skull protects the brain. The spinal column protects the spinal cord. We have some protection around the structures in the pelvic girdle, in the pelvic cavity, thanks to the pelvic girdle there. We don't have any protection for the viscera below the diaphragm, however. The organs in the abdominal cavity are pretty much on their own. They depend on layers of muscle, layers of fat, layers of connective tissue. But all the other structures are protected by bone in some way, shape, or form. Movement. We depend on our muscles to pull on our bones and use the bones as levers to move, because our muscles aren't strong enough to move us. So we use our bones. We use our bones to amplify the strength of our muscles. They function as a lever. And so we can do things. We can sit, stand, walk, run, talk, eat, chew, breathe. All this can take place. We store 99% of our calcium and phosphorus in our bones. And we use that calcium and phosphorus all the time. We have to have calcium for our muscle contractions. We have to have calcium for our nerve impulses. Our heart has to use calcium to contract. So we will always end up braiding our bones if necessary to get the calcium we need. We use our bones as the source of or the location of blood cell formation. We have a structure called bone marrow in the middle of our long bones, and actually in all of our bones. And we start out as children with what we call red bone marrow, and this is where we have hematopoiesis, that is the manufacture of blood cells. We manufacture our blood cells in our bone. As adults, we mostly have yellow marrow because it's mostly fat now, but there are still places in our bones where we produce blood cells. All 7 types of blood cells come from our red bone marrow. Fat storage occurs in the yellow marrow, inside the cavity in the long bones. And we know fat's a great source of energy. It has 10 kilocalories per gram. So we store fat in the marrow, in the cavity, in the middle of our long bones. And you know, if you're used to thinking that insulin controls our glucose levels, you're correct. However, there are other hormones involved too. Osteocalcin helps to regulate the release of insulin from the pancreas. It helps to regulate our blood sugar levels, and it helps to regulate our metabolic activity. So it isn't just insulin regulating sugar, and it isn't just the thyroid hormone regulating metabolic activity. We have a lot of hormones at work here. We're going to talk about that a lot in great detail in AP2. So our bones do a lot for us. They're also living tissue. You know, that's the thing that many of us tend to forget when we're looking at our bones, or looking at a skeleton, or looking at just bone models, that bone is living tissue. It's 40% organic. You know, we tend to think of bone as just a solid structures because that's how we see them. You know, we look at the skeleton, we look at the models, we look at, if we have a real skeleton to deal with, we tend to just view it as an inanimate structure, and that's how we see it when we're looking at, you know, a model. However, the skeleton inside of an individual is living tissue. It has living cells in there because it meets it, you know, it's connective tissue, because connective tissue says living cells in a matrix with fibers. And the matrix in this case is the hard calcium and phosphorus salts, and the fibers are collagen wrapped around the bones, but we have living cells embedded in the bone. 40% of our skeleton is organic in nature. Now we've seen this in lab. We've seen what bones look like. We have flat bones. We have irregular bones. You know, flat bones are like our ribs and our sternum and our skull. Those are all flat bones. They're lightweight, but they're very, very strong. The irregular bones are like the vertebrae. They have unusual shapes, but they have a very distinct purpose for what they do. The long bones, I love this definition of a long bone, longer than they are wide. And that means that any bone that is longer than it is wide is considered a long bone, even if it is one of the phalanges. You know, one of the ends of one of the distal ends of the phalange is a long bone, just like the femur is a long bone. And most of our, a fair amount of our long bones are involved in load bearing, like the tibia and the femur. And of course, we use other long bones for working, like the ulna and the radius and the humerus. So we see these bones here either supporting us, allowing us to move, or manipulating our environment. The short bones, the carpal bones, the tarsal bones in the hands and the feet, and the sesamoid bones. They look like sesame seeds. The patella is a sesamoid bone. And here's what they look like. You've seen this in lab already, so I'm not going to beat this into the ground. Okay. When we look at the bones in our skeleton, bones are organs because an organ is multiple types of tissues working together in a structure to do something. So we have bone cells, we have nerve cells, we have epithelial cells lining our blood vessels, we have fat, we have hematopoietic tissue here, we have cartilage either embedded in the bone or on the end of the bone, and of course we have muscle cells attached to the bone. So bones are organs, and we can look at them from the gross perspective, the overall approach. We can look at the microscopic structure, and we can look at their chemical makeup. Oops, went the wrong way. Okay. From the gross anatomy perspective, bones consist of two types of, these structures are made up of two different types of bone. We have compact bone and we have spongy bone. Compact bone is what we find on the outside of all of our bones. It gives it its smooth appearance. It gives it a solid appearance. On our long bones, the shaft of all of our long bones are mostly compact bone. It's very strong and very powerful. It's also pretty heavy too. Underneath this compact bone, particularly at the ends of the long bones, we have what's known as spongy bone. Spongy bone is lightweight material. It's still bone. It still has bone cells in there, living bone cells. But the spongy bone is designed to be a shock absorber and a shock dissipator, to spread the shock out and eliminate it. Whenever we have areas where we're going to have a lot of stress and a lot of torsion, twisting actions, we're going to have spongy bone, because spongy bone can take that shock and torsion and completely eliminate it and spread it out. And the spongy bone looks like a sponge. It looks like a honeycomb. It looks like you took a sponge and cut it in half. I mean, you can see all the little cavities in the sponge. That's what spongy bone looks like. And it has structures in here that cross-brace it, called trabeculae. We see this particularly at the ends of the long bones. This is a femur down here. And here's the head of the femur at one end and the neck in the middle. And we see the greater trochanter over here, and the lesser trochanter is probably on the other side here. Here's the compact bone running down the shaft of the femur, giving great strength, against twisting and torsion, and support for standing and running and walking along in here. Here is our medullary, what we call the medullary cavity in here. So this is a very powerful bone. We have a thin layer of compact bone on the outside around the head and the neck and the trochanters. Underneath all that is spongy bone. Lots and lots of spongy bone designed to take any shock that comes up the legs and from running and walking and just standing there and spread it out, dissipating so that it doesn't affect us, or what gets transferred up through the acetabulum, through the sacroiliac joint into the intervertebral discs is very minimal. That's what they do. Wherever we're going to have stress and twisting and any kind of shock, we're going to have lots of spongy bone. You'll notice that this is the area also where we tend to see fractures. We see fractures at the neck of the femur. That is the most common hip fracture location, because osteoporosis will attack this spongy bone and dissolve it and leave behind nothing but the outer shell of the compact bone, which is not strong enough, ultimately not strong enough to prevent this from breaking along here in the neck. Now, a person could go for years and just depend on their compact bone, or it could snap at any day, if they have osteoporosis, for example. The average person, a person without osteoporosis is unlikely to have a fracture here because of the bone being so weakened. There's a term here called diploi. Diploi is a sandwich formed between two layers of compact bone with a layer of spongy bone in the middle. And this is what we find in our flat bones and our short bones, our irregularly shaped bones. We're going to have this diploi structure. Diploi structures give us strength without a lot of weight. This is what diploi looks like. This is the sandwich here. This is the parietal bone here from the skull. A layer of compact bone on the top, a layer of compact bone in between, and a layer of spongy bone in the middle. And this is what the spongy bone looks like. This is what the trabeculae do. Shock, twisting, torsion, movement, whatever, is when it strikes this bone, it is spread out throughout the bone, through all these trabeculae, and the shock or the force gets weaker and weaker as it spreads further and further out. We dissipate the force that strikes the bone. And so you bump your head, and it doesn't cause any damage. You bump your head, you may not even notice it when you bump your head. It's possible. Because we're moving our head around all the time, so we absorb the stresses that our head happens to encounter through this spongy bone. The beauty of spongy bone is it gives us great strength without a lot of weight. Imagine how large our skull would be if this was all compact bone. Imagine how much more heavy our skulls would be if that were all compact bone. And think how powerful our neck muscles would be to hold our head up. It would not be unusual, because that's how, if we had compact bone instead of spongy bone in our skull, the norm would be to have a very large, thick skull and have lots of neck muscles to hold our heads up. But we don't have that. We have strength without a lot of weight. Spongy bone does an excellent job here with this sandwich approach. And the construction industry took an idea from this years ago. Bricks, you know what a brick is. Bricks are now made with holes in the middle of the brick. And you go buy bricks at Home Depot or Lowe's or someplace, or at some other supply house, and they all have holes drilled, perforating the middle of the brick, not the sides. And when you build something with these bricks, the bricks are lightweight, and yet they're very strong. They've sacrificed the material in the middle of the brick for the same level of strength from the walls of the brick. And so we build things with bricks that are not, the bricks are not solid. They don't need to be. They give us the same level of strength. The same applies to concrete blocks. Concrete blocks probably 100 years ago were solid. And then somewhere along the way, an engineer discovered that if you hollow out the middle of the concrete block in 2 places, you can have just as much strength as you with half the material as you did with the solid block. And so the idea is that these blocks and these bricks are lightweight. They're lighter than they were before. They're just as effective. The same with our skull. We don't need to have solid bone on everything. We have spongy bone. It makes our ribs lightweight. It makes our skull bones lightweight. It makes our scapula lightweight. The vertebrae are lightweight. Yeah. So it enables our bones to be more efficient when the muscles pull on them. The muscles don't have to work as hard to make them move. Okay. This is a diploid. This is what our sandwich looks like. Here again, there's a parietal bone with the compact bone on the outside and the spongy bone in the middle. The spongy bone functions as a shock dissipator for us, and it keeps these structures lightweight, just as strong, but lightweight. So we're looking at a long bone, a little bit different structure. We have a shaft called the diaphysis. That's the long part of the bone. The ends are called the epiphyses, and the bone is covered in a series of membranes. Now the diaphysis, the long shaft, it has a cavity in the middle of the shaft called the medullary cavity. It can be filled with red marrow to make blood, or in most of us now as adults, it's yellow. It's where we store a lot of fat for energy. The epiphyses, the ends of the bone, are where growth occurred up until about age 23. Those are where the growth plates were located. We call them the epiphyseal plates, and they are where bands of cartilage are located. Because the cartilage cells become bone cells as they reproduce. And so our long bones grow lengthwise at these epiphyseal plates. And they will continue to do so until we reach about 23 or 24 years of age. And then they stop growing. And then they calcify.
Yes. Can you wait a minute?
And they leave.
You handing me over a stupid toy that I told you I would get. I mean, come on, buddy. Well, you don't need it right now.
Okay. Okay, it happens. Now, the epiphyses are the ends of the long bones where we have the growth plates. When the growth plates close, they show up on X-ray as a white line. When they're open, where we have hyaline cartilage present, we have a dark band, because hyaline cartilage is soft tissue, and soft tissue doesn't show up on X-ray. And it looks, here's our epiphyseal line right there. This is the epiphysis, is this end right here. There's the distal epiphysis here. The shaft is the diaphysis. We find spongy bone here at the proximal epiphysis. Our bone grows outward from this line this way and outward from the line down here. Within the long bone, we have the medullary cavity, And in this case, since there is a line here and not a gap, we have yellow marrow. This is what this looks like. And this is an individual that's probably around 21 years of age, I would guess. In the femur, the epiphyseal plate is just about closed. I mean, you can still see these dark shadows in here. That's where we still have hyaline cartilage. Hyaline cartilage is soft tissue, and soft tissue allows X-rays to pass right through it. So we can't use it for identifying structures in the heart and lungs, things like that. So back here. So we see that we're sort of a toss-up between the line is, this is just about closed, but there's still some dark areas in there. When it closes, it leaves a white line, because when the hyaline cartilage stops reproducing and calcifies, there's no more growth taking place at these epiphyseal plates, and they form a white line and say, oh, there's the epiphyseal scar. That's where the growth was occurring. Now, the reason I say this person's probably 20, 21 years of age, if we look at the tibia and even the fibula over here, you can see this dark line. It's not a gap in the bone, it's where the hyaline cartilage is still working. So not completely closed yet, but getting close. So that's why I think it's probably very early 20s on this age. Here we see a child's femur and tibia and fibula. You can see a definite line here. The epiphyseal plate in the femur and in the tibia and in the fibula. But now look at the same structure later on. Now, at post age 23, all we have left is a line across here. All we have left is a line, because the bones, the hyaline cartilage has completely calcified, osteocytes have taken over, and all we have left is this scar. And when you see an X-ray of a long bone from an individual, if you see the dark space in here, and if the age is, if you can't communicate with the patient, if it's the appearance is indeterminate in their age, if you have an X-ray, at least it will give you an idea of an approximate age range. Over here we see, we see the line. We know well, at least they're older than early 20s. So again, this is not, we don't go X-raying people just to try and guess their age, but this is a good indicator as to their overall age. Because we see there's no more once you're an adult, there's no more longitudinal growth, at least there shouldn't be. And our bone cells will continue to grow, and our bones will continue to grow from side to side. They'll shrink and grow based on the demands we place on them. Our bones are going to get bigger if we're doing strenuous activity, if we're a weightlifter, or if we're just doing lots of strenuous work. Our bones, or you're just working out, your bones will get bigger. The bones will lay down more and more bone cells. The bones will get bigger based on the demands that we put on them, or they'll get weaker if we don't have a lot of demands. But that growth is going to be side to side, or the shrinking is going to be side to side. Our bones are not going to shrink lengthwise. They're not going to grow anymore lengthwise. So what does a long bone look like, from a gross perspective? Well, if we're looking, this is an example of the end of the humerus. This is part of the head of the humerus right here. We have the hyaline cartilage that is on the outside surface of the bone. That is the articular cartilage that we have lining the medullary cavity and the head of the humerus. It's to prevent any friction that would occur if those bones were to come into contact with each other. Underneath that, we have the compact bone itself. There is a layer in here, which we'll see in this next slide, called the periosteum, that covers the outside of the compact bone. And its counterpart is the endosteum, which lines the inside of the compact bone. All of the spongy bone and all of the cavities that run through here where blood vessels are penetrating. So the periosteum is one of the two membranes. The periosteum is on the outside of our bone, covers the bone except the joints. It has a fibrous layer that attaches to the bone surface. It acts something like Velcro does, and then it sticks to the surface of the bone. The fibers that attach it to bone are called Sharpie's fibers. They're the ones that sort of mimic Velcro. How Velcro works. They hold this layer, the outer layer of the periosteum helps to hold blood vessels and nerves that are going into the bone, because our bones have lots of openings. We saw the foramens, particularly in the skull. Well, we have foramens in our bones too, and those foramens, called nutrient foramens, allow blood vessels to go in and out of the bone, and nerves to go in and out of the bone, because we have to feed those cells. Underneath the fibrous layer, we have something called the osteogenic layer. The osteogenic layer. Now, when you see the suffix genic or genesis, it means creation. So this is the layer where osteo means bone. This is the area where bone creation takes place. We have bone stem cells here, which will give rise to all four bone cell types that we encounter. And so this is where stem cells are cells that are like an originator cell, a progenitor cell, meaning that other cells come from that. An osteogenic stem cell gives you all four bone cells. A blood stem cell gives you all 7 blood cells, 5 white cells, red cells, and platelets. In the ovaries and testes, the stem cells there give us the sperm and give us the eggs. And so we see, the inner layer in bone is where we have our stem cells here that will give us all 4 different types. And also on the surface of the periosteum is where our tendons and ligaments from our muscles attach to the bones. Because muscles don't pull directly on bone. Muscles pull on tendons that pull on bone. Muscles pull on collagen fibers that pull on the bone, so that we don't damage the muscles when we contract. We don't damage the bones when we contract. We use these fibers, these tendons, to actually pull on the bone. So this is what the periosteum and the endosteum look like. The periosteum is the outside membrane. The endosteum is the internal membrane here. Here is the periosteum with both its Sharpie's fibers here, and underneath that we have the osteogenic layer. Can't see the osteogenic layer here, but it's underneath the Sharpie's fibers. I mean, it's essentially fastened to the bone. We have the opening for the blood vessel. We have the nutrient foramen. The blood vessel will penetrate into the bone, and it will eventually be in contact with every living osteocyte, every living bone cell inside the structure of the bone. And of course, there's the bone marrow. In this case, it's yellow in what we call the medullary cavity. So our endosteum lines the inside of the compact bone and lines if there's spongy bone, it will line all the trabeculae and all of the cavities that blood vessels are going to have to go through. Yeah. The endosteum, is connective tissue. It lines, as I said, lines the cavities. It also contains the stem cells. We have these osteogenic stem cells on both the periosteum and endosteum. And as it says here, they can differentiate, they can become other bone cells. And that's what any stem cell does. It differentiates into a variety of bone cells, a variety of cells. One stem cell in the red bone marrow can produce all 7 types of blood cells. Now, the red marrow is found in the diploid areas. It is in the trabeculae of the spongy bone, and it is in the diploi of the flat bones. And we find as adults that most of our red bone marrow is found in the sternum, in the iliac crest, in the ribs, and in the bones of the skull, primarily in our sternum, but again, also in the iliac crest. If a person is tested for a stem cell transplant of bone marrow, what that involves is taking out a sample of bone marrow and examining it to see if the stem cells in the bone marrow, in the red bone marrow, can match with a recipient. When you hear about stem cell transplants for bone marrow, what we're looking for is to see if those cells in the red bone marrow can work in somebody else. And usually those samples are collected at the iliac crest, because that's where we have red bone marrow. If you've ever had an X-ray that involved putting a lead apron, from your from the top of your sternum down to the inguinal region and over the ovaries or testes, it isn't just to protect the ovaries and testes. It's also to protect the sternum and the ribs, because that's where bone production, blood cell production is taking place. So a lead apron, if it's used, is designed to protect those blood stem cells found in the sternum and ribs. So we're, as adults, most of our blood formation is taking place in some of the flat bones, like the sternum, the iliac crest, for example, some in the ribs, some in the skull, but mostly in the sternum. We do find, though, that if a person develops any kind of blood disorder, a lot of the yellow marrow will gradually convert back to red marrow so that more blood cells can get produced. And this is what our bone marrow looks like in the medullary cavity. Yellow marrow is fat, red marrow is blood producing stem cells. And we find the red marrow, for example, you see here, this is a long bone, there's the spongy bone end at the condyles. Here's the other spongy bone ends up here. Here we see the yellow marrow in the medullary cavity, and here we see the red marrow up in this region here around the spongy bone up here. What does it look like? It looks like this. This is the head of the femur showing the red bone marrow. Because again, the head of the femur is part of this whole diploid type structure. And this is one of the other areas where we see blood cell formation. So our bone marrow would look like this, where we're having blood cell production taking place. Red marrow here, yellow marrow in the middle. So, and the red marrow is where hematopoiesis occurs. The differentiation of the stem cells into all 7 types of blood cells. So we go to the microscopic perspective in here. We see that our osteogenic cells can differentiate into 4 types of bone cells. We have the osteocytes. Those are the living bone cells. Those are the cells that are found trapped in the lacunae in the compact bone and in the spongy bone. The osteoblasts cells are the bone building cells. Think, blast is a builder. Osteoblast. These are the cells that lay down the matrix. For the bones to live in. Yeah. These are the cells that will form the lacunae and get trapped, and some will can become the osteocyte set. The osteogenic cells are made by the osteogenic cells. The osteogenic cells, the stem cells, will always make copies of themselves too, because they want to keep, you have to keep the stem cell line alive. So the stem cells will make additional stem cells. And what happens, like in, for example, in the testes in the male, the stem cells that are going to produce sperm cells will undergo mitosis, and half of those daughter cells will stay in the testes as stem cells. The other half will develop into sperm cells. So we always maintain some stem cells so we can keep producing all the other cells. So the stem cells will give rise to more stem cells. And then the last type of cell that's made is the osteoclast. This is a very large cell that breaks down bone. It has lots of active lysosomes in here, and it will slide across the bone surface and dissolve away the bone and release the calcium into the blood. And so we use our osteoclast when we are remodeling bone. We tear down old bone to make new bone, and we do that every day. We are constantly tearing down bone to build up new bone. We reshape our skeleton all the time. Every 10 years, we've essentially built a brand new skeleton within our bodies. So we replace our skeleton all the time. Osteoclasts do that. It's sort of like one of those large machines you see on the road when it's going along on top of asphalt and just chewing it up as it goes along. It's a large, wide machine. It goes down an asphalt paved road, tears up the asphalt, and throws the asphalt behind it into a container. And that's what the osteoclasts do. So, now. So the osteogenic cells are the stem cells, and they will create other stem cells. They can form osteoblasts. They can form bone lining cells, cells that line the cavities. They can stay as stem cells. The osteoblasts create the matrix. We call that the osteoid. The osteoclasts are very large cells that their role is to break down the bone. And of course, the osteocytes are bone cells trapped in the lacunae that are alive, and they detect changes and stresses in the bone. They tell the bone it's time to grow or shrink. Yeah. This is an osteoclast right here. This is a large cell, because there's an osteocyte right there in the lacunae. But this is an osteoclast. Osteoclast is characterized by multiple nuclei, and it has this ruffled border on the edge here. This is what slides down across the bone and dissolves away the matrix and releasing the calcium. You can compare the size of the osteocyte with the osteoclast. It's an important cell, because we need it to remodel our bone all the time. Other structures we see microscopically. If you remember back to the beginning of lab, when we looked at the various types of connective tissue, and you saw slides of bone, you saw the circular pattern of compact bone under the microscope. We call that lamellar bone. Compact bone, also called lamellar bone, because a lamella is a structure that looks like tree rings. The circular pattern of tree rings are the layers of the rings are called the lamellae, and the entire structure is called an osteon. Now, within the osteon, we're going to have canals, and tiny canals called canaliculi. And we're going to have lamellae that run in the osteons. We're going to have lamellae that run around the outside edge of the bone. That's the circumferential lamellae. So here's a really busy picture for you. Here's a bone right over here. There is the medullary cavity. There's a blood vessel going in through a nutrient foramen. And we see an enlargement of that one region right here. An osteon is like a column that runs the length of the long bone. It's like if you had a bunch of straws together in your hand. Each straw, each straw is about a foot long, so you have 24 straws in your hand, and they're all the same length. Osteons, imagine that type of arrangement inside a long bone. Imagine that this is a straw, and this is a straw, and this is a straw, and this is a straw here. They all run the length of the bone. They run up and down vertically. They're all connected to each other through these blood vessels that run, through here. There are various cavities that allow the blood vessels to penetrate. We call this either the central canal or the osteonic canal or the haversian canal. It doesn't matter, central canal or haversian canal is the classic name for it. But up through the middle of each osteon, up to the middle of each column are blood vessels, arteries and veins, or arterioles and venules, and nerves that are going to be in contact with every little living cell. And here in our osteon, the cells are all osteocytes trapped in the various lacunae through here. And so all these dark spots are lacunae, each one containing an osteocyte. And each osteocyte is communicating to all the others through these connections. You'll notice that this connects to that one, and that connects to this one, and that one, and that one, and even indirectly over to here, all the way up and down the osteon, all the way across the osteon. These cells are communicating to each other. And we have this cluster of columns in the long bone. And we see, these little canals are called canaliculi, and we have similar structures in our spongy bone. Again, there are lacunae. The lacunae are the gaps that form in the matrix where the osteocytes are located. Now so this is the osteon. It is a cylinder that runs the length of the bone. Imagine if you have 20 or 30 straws you're holding together, and they all run the length, they're all the same length. This is what an osteon is. All the osteons are the same length. The osteons, though, are made of layers of bone laid down. Those are called the lamellae. Each layer, each ring of bone is called the lamellae. And the lamellae are laid down with collagen fibers embedded in them. The lamellae are made up of the matrix, the hard matrix of the bone. And the collagen fibers are embedded in the matrix. Collagen, of course, the most abundant protein in the body, stronger than steel strands of the same size. They're wrapped around each ring in opposite directions. So that crosshatch effect, as you see here, we're going this way, going the opposite way up here. This reinforces the strength of the long bone. So our bones are very difficult to break, even though we do. if you've ever had a fracture, you know, bones do break. Of course, the interesting thing about a fracture is you actually never remember the actual fracture. You just remember the consequences of it. So but our bones are very powerful. They resist twisting and torsion very, very well. They can withstand stress, a great deal of stress. It takes a lot of force, a lot of effort to fracture a bone. And so in the middle of the osteon, we have an artery and a vein and a nerve. We have multiple layers here, and each layer is going to contain lacunae that hold osteocytes that run up and down and all the way around. So the canal that runs through the middle of the osteon is called the central canal or the Haversian canal. We have perforating canals called Volkmann's canals that run at right angles. So the blood vessels that come in and go out through the nutrient foramen can then spread and connect to all the osteons in a bone. So every osteon is going to be supplied with blood vessels that bring in oxygen and nutrients and take away waste products, and also supplied with nervous tissue, because they have to communicate. They have to communicate stresses and changes and demands. So the lacunae contain the osteocytes. Remember, the osteoblasts lay down the matrix. The osteoblasts become trapped inside the lacunae and are converted to osteocytes. They can't divide anymore. They can't get out, but they communicate through to all the other cells. As the matrix hardens, it forms these tiny canals that extend from lacunae to lacunae, and the osteocytes extend their cell membrane through these canals and connect to other membranes from other cells through gap junctions. So the communication is very rapid. So, you know, every one of these osteocytes will extend their cytoplasm, extend their cell membrane out through these canals. However many canals there are in a lacunae, they're going to extend their membranes out. And other cells are doing the same thing, and the membranes join in the canals. We have gap junctions that form so that we can communicate instantly cell to cell. And we see there is our osteocyte in its lacunae. Here are the canals, the canaliculi here, and these are the extensions of the cell membrane for each one of these, this particular osteocyte. And it will connect, you know, there's an osteocyte, there's another one there in the lacunae, there's the central canal. And the canaliculi are the passageway for these extensions of the membrane. And they see, let me go back here. And so what you're seeing is there's an osteocyte there and another one here, and each one of those is extending its cytoplasm, its cell membrane out, and it's contacting the cell membrane of this one and indirectly that one, and it's contacting this one, and they have gap junctions between them so they can instantly communicate. They can so that cells are, you know, are in contact with each other all the time. Nutrients can be passed back and forth. Waste products can be removed from one cell to another and passed on till we get to the blood vessels. So, you know, that are needed. Everything is working to provide everything the cells need to be alive is provided through these canals. Yeah. Something else we see. So when you look at this picture here, you can see These are all lacunae running up and down in a circular pattern. You can see the little canaliculi present here. And so we can communicate from layer to layer to layer, from lamella to lamella to lamella. Each one of these rings is a lamellae. This is a concentric lamellae forming the osteon. But if you were to hold those straws in your hand, you would notice that you didn't have a perfect circle. You would have an irregular edge. So to form the perfect circle of bones, probably not perfect circle, but to form the circular shape of the bone, we use lamellae called circumferential lamellae, which forms the outer ring of the bone surrounding all the osteons. And then to fill in the spaces, we use interstitial lamellae. So this is a circumferential lamellae here running in a ring pattern around the bone. And then all these gaps are filled in here by interstitial lamellae. So we have our outer ring and we fill in all the spaces around the osteons that are present. And so we see it here. There's an osteon, there's an osteon, there's an osteon, there's an osteon. There's a conversion canal there and in there and in there. And you can see the lamellar rings here, and you can see all these little lines of the canaliculi. Here are the interstitial lamellae that fill in the gaps. Now they are usually left over from bone remodeling, but we use them to fill in the gaps so there are no spaces between the columns. And then the whole structure is then surrounded on the outside by the circumferential lamellae that give us the nice rounded, smooth surface of our bone. Now, spongy bone is a little different. Spongy bone is extremely important to us. It is the this is the stress dissipating bone. Spongy bone isn't organized in osteons. Spongy bone looks like it's just sort of like the lines look like they're just sort of thrown together, but that's not the case at all, because they are organized in a pattern so that you can spread stresses and shocks out rapidly all through the length of the spongy bone. You know, like if you throw a rock into a pond, you get ripples. And the strongest ripples are at right where the rock hit the water. As those ripples spread out, they get weaker and weaker and weaker. And that's what happens here. The lamellae, the trabeculae, let the shock spread out. Any kind of shock or twisting or torsion that affects that spongy bone, that the spongy bone receives, you know, just like walking. Any kind of force that the spongy bone receives is going to get spread out through the trabeculae till it dissipates, till it's an entirely manageable force that will get spread then. You know, like if it's coming up the femur, you know, what enters the vertebral column will be absorbed by the intervertebral discs and by the pubic symphysis in the front, because we have knocked that force down to almost nothing simply by spreading the force out through the spongy bone. And again, it's like I said, it's sort of like the ripples in a pond when you throw a rock into it. The further away the ripples get from the source, they get less and less and less because they're spreading further and further out. Now, we don't have any osteons here, but we do have sort of an irregular shaped lamellae, and we do have bone cells. We have lots of osteocytes in here connected by the canaliculi, and they are getting supplied, these bone cells are being supplied by capillaries that are found in the endosteum, in the inner lining here. So they work extremely well in protecting our bones from sudden twists and sudden turns and sudden bends. And the only reason we have hip fractures, for example, is it's because all the spongy bone that would normally protect us from a sudden twist or jolt or blow to our hip is gone. Osteoporosis has dissolved the spongy bone, and there's nothing there to protect us except the outer shell of compact bone. And again, when this is gone, we have no protection anymore. Now, chemically, our bones are about 40% organic and 60% inorganic. The organic components are all the cells, the osteoblast, the osteoclast, the osteoid matrix, the osteogenic cells, and the osteocytes. And of course, all the cells that line the cavities too. The osteoid is laid down by the osteoblast. It is the gel-like matrix that hardens into solid bone. It is a mixture of ground substance, which is another term for a gel, and filled with collagen fibers, calcium, and phosphorus salts. And gradually that hardens and gives us the bony matrix in here. This osteoid is what gives us our great strength in our bones, because it's woven through with these salts that harden, and it has all these collagen fibers woven through here too. One of the great strengths that our bones have is due to these collagen fibers. The collagen fibers have what are known as sacrificial bonds. And a collagen fiber will allow itself to be broken. It'll allow the chemical bonds in the fiber to break when the bone is exposed to stress. And by breaking, the collagen fiber absorbs a force or a stress or a twist or a torsion or a blow or something. But the collagen fiber breaks, and the bone doesn't. That's what sacrificial bonds do. They break themselves so that the bone won't break. They allow the force that's being applied to a bone to be dissipated in the collagen that's wrapped around the bone, and then those bonds can reform, and we can break again and reform. It dissipates the energy from the blow or the force without causing the bone itself to break. So that's one of the great things about our bones. It gives us this great strength in here, and yet we overwhelm that all the time. If you've ever fractured a bone, then we know it's possible to overwhelm the collagen, and it happens every day to lots of people. But then there are plenty of times, many times, when we've experienced some sort of trauma. We've fallen, we play contact sports, we drop something heavy on our foot, and we get our hand caught in the door accidentally. You hit yourself with a hammer, hit your thumb with a hammer. It happens to people. We do all sorts of things. We fall off ladders, we fall down stairs, we trip over our own feet. We do clumsy things, and yet we don't break anything. And then sometimes we do. Sometimes things happen. Most of the time we're protected by the collagen fibers and the strength of our bone, but sometimes we have fractures. And it usually takes a lot of force applied in the right spot at the right time for the right duration, which to us is like instantaneous, for a fracture to occur. Now, the mineral part of the inorganic part of our bone, about 60 to 65% are made up of the crystals of calcium and phosphorus wrapped around the collagen fibers. this is what gives our bones their great compressive strength. Bones can resist compression very easily. It makes our bones hard. It makes our bones durable. Our bones will last for hundreds of years after we're gone because of this, because of the inorganic components. We know that bone is half as strong as steel in resisting compression and as strong as steel in resisting tension. So it does take a lot of effort to break our bones. Thanks to all that, the inorganic components, the calcium and the phosphorus, with the organic components wrapped around it. The thing about bone, though, is that bone doesn't break down easily. It takes hundreds of years for bone to break down. A skeleton, skeletons are routinely found at construction sites from old cemeteries that were unmarked. Skeletons identified as far back as revolutionary times, early settlers. It's not unusual every year to read about some Civil War burial ground being found, 150, 175 years old in here. The bones are still intact. And bones will reveal a lot about an individual. First of all, they can tell you how the person died. If there's blunt force trauma, if there's a penetrating injury, particularly in the head, bones can tell you about the nutritional makeup of the individual by what we find when we analyze the bone for organic and inorganic components. Bones can tell you if they were poisoned. Certain poisons like arsenic embed themselves in the bone and stay there for years. Different types of minerals like lead can move, and mercury can be absorbed, and other metals can be absorbed into the bone and stay there for a long time and give an indication as a cause of death. So you can tell by the stature, you can tell that obviously the sex of the individual by the shape of the pelvic girdle, but you can also tell their height and their weight, and maybe even what their occupation was by looking at the bones. Because bones, certain occupations will cause certain, because bones grow in response to the stresses that we place on them. And a lot of occupations with repetitive motion, we'll see changes to bones that last for a long, long time. Okay. Now, term here, osteogenesis. Osteogenesis, the creation of bone. Ossification is another term we use here. When bones ossify, they become calcified and are converted from soft tissue, hyaline cartilage into bone. This starts somewhere in the second month after conception. So usually we say that about 6 weeks after conception, 6 weeks after conception, we have a full skeleton in place made of hyaline cartilage. That's our model. And we'll start laying down the bone at that point, starting at 6 weeks after conception. And this bone growth will continue until we're about 21, 22, 23 years of age, early 20s. And that bone growth will occur up to that point. After that, we engage in remodeling and repair. Unlike muscle cells and unlike nerve cells, we make new bone cells all the time. And we're constantly changing the shape of our skeleton based on the demands. Bones can get bigger, bones can get smaller, bones can get stronger, bones can get weaker, depending on what we're doing. Now, there are two types of ossification that takes place in our bodies when we're in utero. We have what's known as endochondral ossification. This is where how most of our bones form. We use the hyaline cartilage model, and we start replacing that cartilage with bone. The hyaline cartilage cells are gradually converted to bone, and generally this endochondral process, it forms most of our skeleton, and it will continue until the epiphyseal plates close at age 23. Because the epiphyseal plates are made of hyaline cartilage. We gradually replace it. There are a few places where we have what's known as intramembranous ossification. We have fibers. We have fibrous membranes that are converted to bone. They show up in the skull. Bones of the skull, bones of the clavicle. The clavicle bones are fibrous membrane. Just about everything else is hyaline cartilage. But we use both processes here. Now, there's a reason what we have the membranes at the skull, because the skull has to expand as the brain grows. And we have that big fontanelle at the top of the skull, the anterior fontanelle. That's in place to allow expansion of the skull, because our brain gets really big really fast. Okay. So endochondral ossification starts in about 6 weeks after conception. We follow the hyaline cartilage model. We start out with, in our long bones, we have what we call a primary ossification center, where we start forming osteoblasts. Once we form the osteoblast, we can start the process of converting that bone, that hyaline cartilage bone, skeletal structure. The hyaline cartilage bone, that doesn't sound right, that's what's happening. We can convert the hyaline cartilage bone into real bone. So what will happen here? Well, it's a 5-step process. We form a collar, a bone collar around the shaft of the cartilage. We start forming cavities. We create spongy bone. Then we start seeing the formation of osteogenic cells and osteoclasts and osteoblasts. Our shaft and longs, we start having additional ossification in the epiphyses, and eventually we end up with only the epiphyseal plates showing up here. It's a pretty straightforward process. It starts in the middle of here is a hyaline cartilage bone, if you will. Not a real bone, but a hyaline cartilage bone. We have a bony matrix that forms here in the middle that becomes calcified. We start laying down spongy bone. We create a medullary cavity. We have stem cells that are gradually forming the other bones, and we grow. We have this bone collar. We grow out from the bone collar, and we grow out inward and outward from the epiphyseal plates. We end up with that. And this is what we look like. Here we are at 3 months, 12 weeks. Our skull bones are membranes that are gradually ossifying. The rest of the skeleton is endochondral ossification. They are the hyaline cartilage models. And you can see where bone is getting laid down. The dark areas show up as bone. They show up here. This is where bone has already formed, these dark areas, where we can see the phalanges and the metacarpals, and we can see the ulna and the radius. Now, there are no gaps here. What we perceive as gaps are the areas where we still have hyaline cartilage, the femur and our tibia, and our part of our pelvic girdle in here. We can see the ribs present in the scapula, and our clavicle looks like it's almost fully formed. All of these bones, the whole structure is intact. Where we see the gaps is where we have hyaline cartilage. We don't see the the bone has not laid down in that cartilage yet. Like here's the sternum. You know, we see these gaps in the long bones. Well, that's because there aren't really gaps. It's where the cartilage is still present. So this is at 12 weeks. This is 6 weeks after calcification has started. So, and we're probably a couple inches long at this point. We're not very big. We started the process when we were 1 inch long. We're 2 or 3 or 4 inches in length at 12 weeks. So, and again, here we see this is, I think this is about 10 weeks right here. We can see because there's less bone development than we have here at back here at 12 weeks. This may be 10 weeks, but we see the long bones are forming, the ribs are forming. The pelvic girdle isn't as pronounced yet. The bones of the legs and feet aren't quite as pronounced, but the model is there. Now the intramembranous ossification is taking place here. The bones of the skull, frontal, parietal, occipital, temporal, and the clavicle. Those are going to form from these membranes at the same time that the hyaline cartilage model is being laid down. And with ossification in the membranes, we just start out with ossification centers forming in the membrane and becoming osteoblasts. They lay down the matrix calcifies. We form trabeculae, and we start replacing the membrane at that point. And again, it takes 20 some years till everything is finished. And this is intermembranous ossification. We start out in the middle of the membrane. We form osteoblast. We lay down the osteoid. We trap osteocytes. We form spongy bone from that. And then we end up with compact bone on top of that. So, last point for today. Our long bones are going to grow lengthwise at the epiphyseal plates. You know, we're going to have vertical growth up and down in the long bones where the epiphyseal plates are. When they close, we don't get any more vertical growth. We've reached our adult stature, or whatever stature we're going to be. Thickness occurs what we call appositional growth, where we grow from side to side. That will continue throughout our lifetime. So the only other bones that will continue to grow are our facial bones, and that's a very slow process over our lifetime. So our facial bones may continue to grow as we transition from infants to elderly. And I'll stop here at growth and remodeling, and we'll pick up with this on Monday. So, any questions on anything? Okay, then I want to get us out of here, and I will see you all later. Lab this afternoon is on the skeletal muscles, and I sent everybody a list in lab of all the muscles and structures that we're going to be looking at. Everybody should have received that list already, and we'll talk more about that this afternoon. So if you're in lab, if you haven't been in lab yet, lab today and lab tomorrow are going to be the introduction to skeletal muscles. So.